Okay, Chapter 9, Part 2 Helper T cells. Let's talk helper T cells. So, here we have our standard picture. T cell receptor is these two bits, and all these other parts are CD3. Yeah. You'll find CD3 with a T cell receptor pretty much all the time. I think some components of it are absent on T regulatory cells. So they would be able to bind but not activate. And that's part of what would make them regulatory. So they're going to send out negative signals. Yeah. All right. So let's talk. So there are a bunch of different helper T cell subtypes or classes, I guess people call them. T helper 1, 2, Th17, T follicular helper, which are Th2s that hang out in the follicle all day. Yeah, frolicking in the follicle. And T regulatory cells that just tell everybody shh and no. Little librarians of your immune system. Um, so each of these is able to influence separate parts of the body. Now there's some crossover between these. So imagine Venn diagram, I don't know. They might overlap by that much. Yeah. But there are going to be specific things that one T cell type is really good at and the others are just not as good at. So we'll focus on that. We'll pretend that's what they do all the time, 24/7, even though there's a lot of noise in all of this stuff. Yeah, there's a little bit of a lot of things happening at once all over. So Th1 cells are in large part there to influence macrophages. And they can actually shift macrophages to different types. So in the modern interpretation of immunology, some macrophage specialists will classify macrophages as M1 macrophages if they're the kind that are like you get, like the ones that interact with Th2. So interferon gamma, interact with Th1. Sorry, I said that backwards, but you can see it on here. So Th1s make interferon gamma that can activate the macrophages, and then the macrophages can release a bunch of signals, and there's further steps beyond that, but we'll stay focused on the narrow for now. Yeah. So some people would call those M1 macrophages that are interacting with the Th1 or that have been stimulated by interferon gamma. There are M2 macrophages, and some people would say maybe we should say that there are M3 macrophages, but there's not as much consensus there. But that's the idea. And M2s would be different from the ones you would get with a Th1 stimulated response. Yeah. They're like what you would get with Th2, but it's not the helper T cells that are directly stimulating them, it's other cells. So they end up doing different things. Yeah. It's just like imagine a bunch of computers, they're receiving a certain input, they give you a certain output, and the M2s are receiving a different input, giving a slightly different output. There's still a macrophage, there's still only so many things they can do, which include eat and dump out a whole bunch of nasty little things, nasty little surprises. Okay, Th2 cells, they're going to release IL-4, also a bit of IL-5. That's fine. IL-13, don't worry about IL-13. So some of the major targets of this are going to be in the bone marrow. You're going to have some B cells hanging out there. But also the IL-13 would be stimulating goblet cells to secrete mucus. How wonderful. So this would be on your mucosal membranes. And mucus is just this long sugary chain, basically sugar-coated chain. And a lot of viruses and bacteria have enzymes that specifically bind onto those sugars because they're sugars you would normally find on the surface of the cell. Mucus is a decoy. It has the markers that you would find on the surface of the cell. Things like sialic acid is on most proteins that are glycosylated, and most proteins on the surface are glycosylated. So yeah. I believe mucins are just loaded with sialic acids and sugar chains. So they're just mimicking, and it's just like spamming out. It's like when fighter jets will throw out this sort of like confetti to distract heat-seeking missiles. It's the same sort of idea. Yeah. So that's what mucus is. Yeah, now you know. Yeah. Th17 cells. Now these are our sort of early ones, and Th17 cells do not do their thing for very long. They usually get shut down fairly quickly, but in some diseases like SARS-CoV-2, they can have a very, very strong response. Anyway, they can release IL-17, which can activate stromal cells to make some other cytokines and chemokines that'll eventually go in and fire up neutrophils. Th2 cells, now they're going to be mostly about interacting with B cells, but also they can release some factors that are going to activate eosinophils and mast cells and basophils, which are cells from the immune system that are patrolling mostly. They have a very limited ability to present via class 2, only when they get signals from a fired up Th2. That's what basically turns them on and makes them start to present what they've got. But they're not great at phagocytosing anything, so yeah, they probably haven't eaten much. They're not a whole lot of help, but it's like when your kids help you cook dinner or something. It's yeah, I guess it's help in the nominal sense. So fine. Neutrophils, though, very important. Neutrophils, I believe you may have already covered, but they're just the most amazing little selfless assassins. They swarm on anything that's hurting you. They'll rip themselves apart and basically form a net out of their own DNA. It's like a ninja assassin that strangles you with his own intestines. It's just really ridiculous levels of devotion in those neutrophils. And there are a lot of them, and they're pretty effective. They do a pretty good job at just destroying stuff. Yeah. And they'll consume things, but they're not great at presenting. Yeah, they can do a little bit, but mostly they're just there. There's some muscle. There's some cheap muscle. Yeah. T follicular helper cells are going to be the Th2-like cells that mostly live around the B cells, kind of orbiting around them and stimulating them, give them a little extra inspiration, a little extra, you can do it sort of mentality. They will excrete IL-21, and that's one of the things that will cause B cells to differentiate and cause B cells to start to do isotype switching. Like it's one of those little pats on the back that you can give to a B cell is IL-21. And there would also be, IL-21. And then you got T regulatory cells, the cells that say no. So they'll release some interleukin-10. They can inhibit dendritic cells from releasing their various cytokines. And they can basically turn off activated T cells or make it more difficult to activate T cells. Usually once a T cell gets activated, it doesn't last that long, and it'll burn out unless it activates more T cells. It's like one of those chain letters that only goes as long as people keep forwarding it to someone else. Yeah, by the way, forward this lecture to 6 friends to enjoy, I don't know, prosperity or something. Don't do that. Okay. And of course, each of those helper T cells, this is like a different version of that graphic that I showed before. I like the one before because it shows like what makes them work and then a little bit about what they do and then what they produce, but whatever. Yeah. Different form as more or less the same thing. So we've got our various types of effector helper T cells that can actually produce something and stimulate some other cell and get stuff done. And so remember, interleukin-12 and interferon gamma are the 2 signals that it takes, and they work better if they come in together to activate a Th1 cell. Activated Th1 is going to produce a whole bunch of interferon gamma. So if somebody else is producing interleukin-12, then you'll have the key to set off more T cells. So this is like a safety mechanism. This T cell can't directly activate a bunch of other T cells efficiently unless there's another cell type that's producing a bit of IL-12 that says, yeah, I see it too. We both see it, and so then they'll the other T cells can then respond. So it's nice. It's a way to keep ourselves from destroying ourselves. Yeah, immunologically. IL-4 makes Th2 cells, and they then produce IL-4 and IL-5. And you need a little bit of TGF beta. A little IL-6 will help, and definitely some IL-23, and then you'll get your Th17 cells, which happily start producing interleukin 17, which is then going to go out and unlock some of those epithelial cells and get them to do a little bit of class 2 presentation and stuff like that. Secrete some other factors. So all in all, these are helper T cells are mostly there to switch on other parts of the immune system. They coordinate other parts of the immune system by basically being the key that turns them on. They're like the like the little 4-star general, or like the boss. Yeah. Dendritic cells have a lot of this function as well. They can both present antigens and send out a lot of signals, well done signals. And so you can look at dendritic cells as another sort of controller of the immune system, I would say. It's not all helper T cells. Yeah. Okay. IL-6 is one of the things that you need to make a T follicular helper. They will produce some IL-4. It doesn't show it here, but they totally do. And they'll also make some IL-21, which is our signal to the B cells to keep on trucking. Yeah. And then you need transforming growth factor beta and IL-2 together, like the two little keys to the nuclear codes, to unlock T regulatory cells, which will then spit out TGF beta, which will, with other signals, could get us started again, get us cycling to Th17, which gives rise to Th1, which then gives rise to Th2, and follicular helpers come basically at the same time as the Th17s. But unless there's a second signal, they are not going to activate Th17s, so I'm not going to cause them to differentiate that way. And then IL-10 is just a general sort of shutdown sort of marker. It's generally considered anti-inflammatory, although with all these things, there is one cell type or subtype somewhere that will get stimulated by each of these cytokines that shuts down the rest of them. So we're going for broad picture here, not exceptions. Like, learn the rules first, then we'll start layering exceptions on basically after this class is done, I would say. If you were curious, and I'm not going to test you on this, but maybe you are curious, this is how it works. So interleukin 2 is going to interact. So all these proteins, all these interleukins, you see they interact with a common kind of protein called a STAT protein. STAT proteins are signal transduction and transactivation, if I remember correctly. And these are little transcription factors. So they would love to go into the nucleus and bind to the DNA and basically act like a little bookmark and say, they'll bind to a specific spot and say, here, start making RNA right here, please. And that's how cells know what proteins to make in response to a particular stimulus, what genes to express. It's based on where all these different transcription factors land and call in the transcription machinery that polymerase II. So there's a whole bunch of stats. You better believe there's a whole bunch of stats. Interferon gamma is the big one for STAT1, but they also interact in combinations. You'll have STAT1 and STAT2 interacting together. That'll bind a certain sequence. Two STAT1s will bind a different sequence. STAT4s will bind a different sequence, and various combinations. People are still figuring all these out, but they've got some of them down. So all these interleukins bind to interleukin receptors, which are all fairly similar. And interleukin receptors have ways of interacting with STAT proteins and phosphorylating them. The thing that they haven't said here is that there's usually a kinase stuck to the bottom of the interleukin receptor. One of the common ones is called JNK or Janus kinase. So, not pictured. JNK. interact with interleukin receptors and phosphorylate them some stats. Yeah, so that's like the intermediate step. That's like where this arrow is. It's not always Janus kinase. Janus kinases are responsible for this pathway for sure. And there are kinases that are similar to that involved in all the other pathways. You can believe it. Anyway, those will then turn on particular genes. Now, these are interesting genes. GATA3, this is something that gets turned on really early in developing embryos, like when there are only a couple of cells. FOXP3, another key early developmental gene. So basically, these early T cells are kind of like stem cells. And so they're going to turn on some of the same genes that were turned on during early development. And so they literally will develop, on a single celled level from a naive T into a T regulatory cell, T follicular helper cell, etc. It is, it's like a little metamorphosis that happens just at the single cell level. It's going to change the proteins it makes, change its whole outlook on life, and change what it does. Yeah. So that's the power of T cells. They can go from basically inert to all kinds of different things. And the same sort of TH0 cell can do all those. Because a TH0 cell is making T cell receptor, bit of CD3, and a bit of CD4. And that's it. That's all it takes to be a TH0. And then boom, you get the right signals, and look at all the things you can become. Yeah. All right. So this is a dendritic cell. This is how they always draw the dendritic cells in this book, which is nice. Yeah. And they always keep the colors the same as well. So helper T cells are always going to be blue. Naive T cells are always going to be green. T regulatory cells are going to be this ugly sort of orangey, whatever it is. Not quite salmon. I don't know what you call that color. Maybe it's your favorite color. It's a very nice color in that case. So, it's just saying that toll-like receptor activation is one of the things that can set off a dendritic cell and cause it to start having effector functions and cause it to start producing interleukin 6, for example. Dendritic cells make interleukin 6. Interleukin 6 causes naive T cells to turn into both actually T regulatory and Th17 and T follicular helper cells. All those pathways would potentially be affected. As long as they're getting a little transforming growth factor from somewhere else. And if they get a lot of it, then they'll become a T reg cell, and if they only get a little bit of it, they'll become a Th17. That's what the diagram means when they show these particular pictures. So it's kind of a neat book in that the diagrams are all sort of linked together, and they all use a common strategy and a common set of colors and shapes to do a thing. It's unusual among textbooks, I think, and good, in a good way. Yeah. Okay, so we've learned how cells can differentiate. Now let's learn how cells shut down the differentiation of others, because of course it's a feedback loop. You got to have one side stimulating, one side inhibiting. Get your yin and your yang together, and that's how the immune system normally doesn't go completely off the rails. It's a balance. It's an equilibrium between helping and hurting each of the cell types onto the others. So start over here. Th2 cells love to make interleukin 4. We know about interleukin 4 because it shuts down Th1 cells. And so it's an interleukin. It's going to bind to an interleukin receptor, which looks much like the others. Slight differences in these that make it bind to IL-4 better than IL-6, 7, 8, 9, 10. Take your pick. But otherwise, pretty similar, I believe. Whatever. Go prove me wrong. I know there are exceptions. I just know that they're not particularly common, the exceptions. Th1 cells produce interferon gamma. Interferon gamma shuts down Th2 cell differentiation, so they're not allowed to proliferate. They get deactivated potentially, or makes them harder to activate. And it also is going to inhibit Th17. So IL-4 will inhibit Th17s and Th1s. So look at this. First thing you make are probably going to be these Th17s. And the Th17s, that's fine. They can go out and do things. But as soon as you start to make either a Th1 response with interferon gamma or a Th2 response with IL-4, then both of those things are going to shut down the Th17s. Like I said, Th17s are there to just hold the phone. They're the opening act that nobody really wants to see. They're just filling a space that was there and needed to be filled. And then as soon as the big boys come in, the big girls come in, then Th17s can get out of the way. Yeah. This is the current understanding. I bet in 10 or 20 years there'll be other things that Th17s do that we didn't realize now. But for now, this is where we are. So, yeah, it's immunology, always changing. All right, I don't know what else to say about this one. And then T regulatory cells, neat, can make transforming growth factor beta, which stimulates things like Th17s, but shuts down Th2s and Th1s. So, yeah, each of these things, it's like the three branches of government, all having sort of it's like paper, scissors, rocks, honestly. Each one beats another one and loses to another one. So Th2 beats Th1, but Th1 beats Th2, but Treg can beat both of them unless Tregs get shut down, and Tregs will kind of start Th17s, which are then going to fire up either Th1s or Th2s, which will then, shut each other down. So in any immune response, you're going to have a little bit of all of this happening at the same time. It's not like one thing is on and the other things are all off. It's that one thing is a little more on than the others, and then, there's sort of a balance between them. And it goes back and forth. That's the idea. Yeah. All right. How's this useful? Well, this is a cool little experiment. So Leishmania, fine. I think it's bacterium. Yeah. Who cares? Bacteria. They're not as cool as viruses. Face it. Yeah. Sorry. But not sorry. Not sorry at all. So with viruses and with most live bacterial infections, a Th1 response will save your life. If you make a strong Th2 response, it'll shut down the Th1 response that would save your life, and you'll die instead. You'll die full of antibodies that probably don't do as much good as they ought to, because the rest of the immune system is not primed and is not there, and that's not where you want to be. That's not a good place for anybody to be. So if you inject a mouse with Leishmania, if you give a mouse Leishmania, it'll die. It'll die about 2 months after you infect it. Good job. Yeah. Feel better about yourself? You shouldn't. Yeah. If you give Leishmania, but you also give them an anti-IL-4 antibody. Now think about that. So IL-4 is the thing that's made and produces Th2s, and also made by Th2s to make more Th2s. If you put in an antibody that binds to that so it can't do either of those things, then you're making a mouse that's basically like an IL-4 knockout. It can't make it, and it can't really respond to it because it didn't make it. And so those mice have only one pathway left, one major pathway. They'll start with a bit of Th17 because it always does, but then they're going to go to Th1, and they're not going to switch to Th2. They're just going to stay in Th1 response the whole time. And that will clear the bacterium. And so these mice all survive. That's the idea. So, yeah, but there will be times. There are times when a Th1 response will save your life. There are times when a Th1 response that keeps going will kill you. And there's no way to tell your immune system which time it is. Or rather, the immune system has to figure this out from a bunch of different cells sending potentially conflicting signals to each other. And it's just like a tug of war. Did we pull it a little more this way, a little more this way? And yeah, that's how these things go. That's why The immune system is kind of complicated. That's why it's hard to say, oh, I gave you this vaccine, and therefore you will develop precisely this number of antibodies by exactly this day, and the antibodies will last for, let me check my watch, exactly this amount of time. Yeah, because you can't. Because you got all these different T cells in there and all the other cells in there, and it all depends how each one of these interacts with all the others. That gives you the total state of what's happening, state of the immune system. Yeah. Okay, a little throwback here. So these are our activated CD8s. You can tell because they're this sort of nice dark purpley color. And so look, there's a CD8 coming in, and oh, it's attaching on with its LFA1 to ICAM1. Fascinating. I heard about this. If it attaches and is able to make a contact with its CD8 and its T cell receptor and bind to an antigen that's presented on MHC class 1, and if it's an antigen that it's already learned to recognize once it's activated, then the activated killer T cell will kill. Now it has to get activated first. Antigen presenting cells activate T cells. That is their function. They hold out a little piece of a thing. They say, if you can recognize this, you get the reward, which is you get to be activated. And you get to make more copies of yourself, and you get to go out and do stuff. Otherwise, if you can't recognize this, nothing. So you can think of antigen presenting cells as something like teachers. They're showing a piece of information to this giant swarming class of T cells. And if the T cells are able to recognize it, oh my gosh, is this hitting close to home? Yeah, I bet. If they're able to recognize it, they not only get to live, but they get to prosper. Yeah. If they can't recognize it, yeah. Go away. We have no use for you. Not actually in teaching, but that's how the immune system works. So there you go. Okay. What is this point of contact between the killer T cell and the antigen presenting cell? So we've shown it as a dendritic cell here. And it's actually called a SMAC, supramolecular, meaning involving like how molecules fit together, in this case how proteins fit together. Activation complex, so it's giant. It involves a whole bunch of different proteins all interacting, like exchanging their little, secret handshakes back and forth, and causing T cell activation and signaling and what have you. So it's actually got two zones, because why not? Why shouldn't we subdivide a normal thing? It's got the central SMAC region, which has the actual like T cell receptors and CD4s and stuff like that. And it's got the peripheral SMAC region. This is where you're going to find your LFAs. You'll find your ICAMs if we're talking about what's on the dendritic cell. Yeah. And some other stuff. And proteins will wander in and out of these zones, but they tend to be concentrated there enough that you can actually see it by fluorescent microscopy. As shown here. So here's our little SMAC. So here's proteins just sort of distributed around. And then here we have a clustering of these proteins. I don't remember which one. You'll have to look in the book for which protein this is. It might be T cell receptors. Or it might be MHCs or something like that. And so they're all going to be clustered at this site. We're going to call them in together. And then if, and only if, the T cell is able to make a nice bond with the T cell receptor to the MHC class 1, then fine. We're going to activate the T cell. It's going to release some of its little chemicals. Here's a T cell releasing a little packet of all kinds of good things, really. Cytokines, chemokines, and etc. And they will then have effects on that cell. So that's the idea. Yeah. Okay, so what are the weapons that a killer T cell has? Well, I mean, you've seen Fortnite. You've seen all the weapons in there. This is the arsenal that you get for CD8 T cells. So pay attention. Perforin, shaped like a banana, kind of looks like complement 9, the C9, kind of works the same too. Banana shaped, slots into the membrane. You get a whole bunch of them that bind onto each other. And once they've done that, you basically have a little pore. And all the guts of the cell leak out. It's not any smarter than that, I'm afraid. Granzymes and granulysin are different wonderful ways to kill cells. Fas ligand is going to interact with some of those proteins and force a cell to kill itself. And so that's great. A cytotoxic T cell can just make some Fas ligand, put it on the surface, and anytime it's able to interact with an MHC class 1 that's already been activated and is able to bind, then Fas ligand is going to come in and say, apoptosis time to that cell. And it works really well. Yeah. Then these CD8 T cells will also produce a bit of interferon gamma. So CD8s will drive more Th1 responses, which will drive more macrophages, which can potentially drive more CD8s around in a little circle like that. And NK cells, they'll hop in, and they're part of the circle too. They respond to interferon gamma as well. LT alpha and TNF alpha are other things that killer T cells can make. These are going to go out to other cell types, and they're going to affect their function. It's just like a always ever-growing list of different messaging chemicals, different proteins they bind to, and the effects that they have downstream. That is immunology as she is studied, and understood at the moment. Okay, that was CD8s. What about CD4s? Well, there are all these different kinds of CD4s. So we'll assume that T follicular helpers are going to be Th2-like enough that they'll do things kind of like this. Th1 cells, their big one is interferon gamma. Do you need to know every single one of these things? Mostly no. Interferon gamma, that's worth it. Fas ligand, that's worth it because that's going to set off apoptosis. And I don't know. If we need these others, we'll pick them up. GM-CSF and TNF alpha do come up occasionally, as does CD40 ligand. That's probably one worth mentioning as well. As a potential macrophage activator, but also activator of other cells. We'll get to that. Let's see. Th2 cells are going to mostly make IL-4 and a bit of IL-5. And they'll also make some CD40 ligand, as will Th17. And you see the one cell there's some overlap here, like CD40 ligand made by all three of these sort of more effector helper T cell types. For the T regulatory cells, no. They're going to make TGF beta, which is like half the signal you need to make a Th17. And they're going to make IL-10, which basically calms down your Th1 and Th2 responses. That's how it works. Okay. If you're ever interested in all the effects of all of the different cytokines on all the other different cell types, this is a shortened, very much simplified list. By the way, I think the important things are the red boxes. These are like major functions. This is like the big stuff. The little stuff in the white boxes totally happens. So if you're looking for exceptions to the rule, all the white boxes give you that. The red boxes are more like the rule. So IL-17, mostly in these sort of hematopoietic stem cells, it's going to stimulate neutrophil recruitment. So they're going to be recruited to the site. IL-17 is like it's like a little trail of candy, and they're going to follow it right into the site. So that's how Th17 cells call neutrophils to them. It's like having a secret bird call or something, only it's IL-17 and it's a neutrophil call. So there you go. And then they're going to stimulate fibroblasts and epithelial cells to make more chemokines, and potentially also start to present some antigens. Right. What else have we got? IL-2. That's the big growth factor. So when a T cell gets IL-2, it's going to make some more T cells. It takes two to make a thing go right. Macrophages are going to be activated by interferon gamma. And they're going to start making just a whole bunch of presentation. They're going to present everything that they've got. They're going to turn themselves inside out, not literally, but kind of, By presenting things, they're going to do those things where they crash the lysosomes into other parts of the cell, just in case, present a little bit of everything. Lymphotoxin A, which is lymphotoxin alpha, LT alpha, is going to activate macrophages as well. I think as far as what we're going to cover, I think if you know the interferon gamma does that. I don't know. LT alpha, I might put that as a second choice, which is also correct as a way to, activate macrophages. Let's see. And TNF alpha is also going to activate macrophages. So these are pro-inflammatory cytokines is one thing that they're called. Let's see. Interleukin 4 is going to help. It's going to be a growth and survival signal to Th2 T helper cells in particular, but to some degree to other types. Although it's going to inhibit Th1 helper T cells and Th17s. Let's see. B cells, if they get interleukin 4, that's going to stimulate them. They're going to class switch, and they're going to present, and yeah, they're just going to do their best B cell impression. Let's see. Interleukin 5 is going to be an eosinophil, pro-eosinophil factor, so they're going to grow and differentiate and get all fired up. Interleukin 13 is our snot-making protein, make that mucus. Interleukin 17 we already covered. It's think of it as the neutrophil call. Forget about stimulating fibroblasts. I think the neutrophil call is more important. And interleukin 22, which is also going to be made by those Th17 cells, is going to be a stimulation to make some of those antimicrobial peptides. Think like defensins, think like cathelicidins, stuff like that. Stuff from way back at the beginning of immunology. Do you remember those days? So long ago. TGF-beta is going to help us make Th17s and Tregs. Yeah. And it's going to shut down Th1s and Th2s. That's what it does. So this goes back into the getting ready phase, and then Th1s are in the let's fight now phase. Th2s are let's get ready to fight next time phase, basically. But all those phases are on a little bit, at least, all the time. Let's see. And TGF-beta is going to inhibit growth, of B cells. But if you get enough TGF-beta, that's something that can make some B cells switch to IgA production. So putting out TGF-beta will make you make mucosal immunity with antibodies, and that'll help. Yes, TNF-alpha is going to activate macrophages. We already covered that. IL-10 inhibits a whole bunch of other inflammatory cytokines. So like TNF-alpha, interferons, variety of inflammatory interleukins, all going to be kind of inhibited by IL-10. So IL-10 is like a stop sign. Yeah, think of two hands saying like that, no. Yeah. And think of IL-5 as like one hand saying, come on, eosinophil, come on, you can do it. Yeah. Sort of like that. Yeah. And don't worry about the GM-CSF. Don't worry about it. Yeah. Okay. Killer T cells bind to infected cells via peptides presented on class 1. Once the CD8 T cell is activated, it does not need a second signal in order to kill. So an activated T cell, activated killer T cell is good at killing all by itself. It does not need your help. It does not need your friendship. It is here to do a job. And so it'll just go along. There can be a whole row of cells, and it'll interact with one, send it a signal through Faz ligand, and cause the cell to kill itself, or release some, granzymes, or release some perforins, and kill the cell directly. And then it can roll to the next one and kill that, and roll to the next one and kill that. That's just how they do it. All right, what have we got here? So, this is the this is what happens in apoptosis. It's a super simplified version of apoptosis. If you're in a cell biology class, oh my gosh, you probably had to learn your entire caspase cascade. So it's going to start with 9. It goes like a countdown: 9, 8, 7, 6, 5, 4, and they're kind of activating each other in that way. So there are many different proteins that have caspase recruitment domains. So that's what this card domain is. It is a caspase, Ca for caspase, R recruitment, D domain. When this protein binds to whatever it's trying to bind, so these are like recognizer proteins. If they see something like a viral marker or something like that, there are a bunch of these different proteins. There are a lot of ways to set them off, but when they get set off, they all do the same thing, which is they go from curled up like this to straightened out. And when they're straightened out, now the card domain is available to bind to caspases. And so open card domains can recruit caspase 9, for example. And caspase 9 could interact with a bunch of things, including activating caspase 3. And caspase 3 will go in, and caspase 3 can activate some of the other factors that will go in and break up the DNA and just murder the cell, straight out murder it. So that's like what happens after Fas ligand binds to a cell. Because cells will be expressing Fas, and this T cell comes along with Fas ligand, which binds to it, and boom, when you press that button, that's like the self-destruct button for a cell. Yeah. All right. So there are a bunch of these apoptotic stimuli. They're going to come in through these proteins that have what are called death domains. And there are going to be a lot of proteins that are just sitting on the surface of a cell watching, or sitting floating around inside the cell in the case of P53. And they're going to be trying to detect, is there something here? And so these would include a lot of things that have card domains, caspase recruitment domains. Now, they would inhibit the protectors. So these are like the BCL proteins, Becyl2, Becyl XL, and Becyl W. They would inhibit the executioners, which are actually going to go in and kill the cell. And so you have a way, it's like a little circuit that can be turned on or turned off. So if you make the right sentinels, they'll activate the executioners that will then cause cell death. Make the other sentinels, they'll activate the protectors or they'll stop inhibiting the protectors, which will then be able to inhibit the executioners, which will stop the cell from dying. That's the idea. Okay, what are some of these products? Perforin, yeah, we know about perforin. That's the one that is going to help deliver contents of granules into the cytoplasm, and it's actually going to insert itself in the membrane. Granzymes have serine proteases, which will activate some of these other things and cause apoptosis, and granulysin can also potentially induce apoptosis and have some antimicrobial functions. So I think that's probably, let's see, how many more slides do we have in this one? I think that's a good place. Oh no, that's a good place to finish up this set. All right, 2 more slides and then we're done with this section. So CD8 T cell has got these little cytotoxic granules. It has your granzymes and your granulysins in it, and your perforin. And they are going to perforate the cell, allow other things to come into the cell, and release these activators of apoptosis. And so once those granzymes, those proteases come in, they can then activate procaspase 3 into caspase 3. And caspase 3 can cut the little red inhibitory domain out of CAD. And then once that's done, CAD can go in and just start destroying DNA. That's the idea. They can also activate BID, which is going to interact with BAD and BAX, BID, BAX, BAD. And then together, those are going to basically puncture and cause cytochrome C release from the mitochondria. Yeah, all of which are involved in cell death. So here's that same old picture from before, just showing you with actual cells doing it over here. So the interaction happens pretty quickly, and then the cell signals are sent pretty quickly, and then it takes a little while for apoptosis to take effect. So that's about 40 minutes after binding. But yeah, it's this killer T cell interacting with the infected cell, and then moving on, doing it again. Yeah, to kill again. There you go. If a cell is presenting the same peptide from the same virus, bacterium, whatever. All right. That's enough immunology for today. Thank you very much. This has been Chapter 9.